first webinar of our new series for spatial metabolomics with METASPACE. So we've had a webinar like this last time I think in March last year with the best practices, and it's been a long time, so we thought it would be nice to start revive those webinars and start from the very beginning, which is an introduction to METASPACE. So if you already know METASPACE and know how to work with it, I'm sure you're going to learn hopefully new stuff. And if you don't know METASPACE, then welcome. I'll start introducing everything slowly. So let me first start sharing my screen. Right, I hope you can see my screen. Lucas, is it working?
Yes, it works.
Okay. All right, so let me first start with the housekeeping rules. So this is just so that we are all on the same page. So for this webinar, if you want to ask any questions, you can use the Q&A button down below. You just ask any question throughout the session, and we'll answer it either during or after the session. There will be some checkpoints. And also, please keep your mic muted throughout the webinar. And the webinar is recorded and will be available on YouTube after the webinar, and we'll post it as well to you guys. So METASPACE, before we start, this is our team. This is the team behind METASPACE. So since November 2025, I don't know if you have heard or not, but METASPACE as a platform has split into two different platforms. The first is METASPACE Academic, which is maintained by the Alexandrov team at UCSD, and METASPACE Pro, which is commercialized by the company MetaCloud. So METASPACE Academic is the platform dedicated for public datasets submitted to METASPACE, which is mostly covered by grants. And METASPACE Pro software is only governing the private datasets. So MetaCloud is the company located in San Diego. It's registered under agreement with UCSD to commercialize METASPACE. And this is providing the METASPACE Pro subscription-based cloud software. And we will talk later about this, but this is the main team behind METASPACE. So Lucas, you've already seen, is the CEO and the software development. I'm currently handling the data science, and Theo, I think everyone probably knows Theo, who's the main brain behind METASPACE, and he is the team leader in UCSD maintaining this. So as a show of hands, maybe to get an idea of who here knows about imaging-MS. I'm suspecting that everyone knows imaging-MS, so maybe I'll ask the question differently. Who's not familiar with spatial metabolomics or imaging mass spectrometry? Can you just show like make a reaction or something just to know how many doesn't know spatial metabolomics? Okay, I think most of the people here know. All right, so I'll make it quick. I will not dwell a lot on the introduction, but basically for imaging mass spectrometry, this is the technique that enables spatial metabolomics or investigation of metabolites, lipids, and drugs at the high spatial resolution. So one of the main methods, which is the enabler for imaging mass spectrometry, is called multi-imaging, is the most popular method. Basically, you have the tissue on a slide, and then in multi-imaging, since multi is the ionization source, you shoot a laser on the tissue, on the slides, and then this laser moves in a, for example, rasterized fashion throughout the whole slides, creating these pixels, and in each pixel, you have your own mass spectra, which is the mass M over Z and the intensity. We call usually the data set that comes out of imaging mass spectrometry as a hyperspectral data sets. And the data, we call it as a data cube. The reason why we call it as a data cube is because you have your normal spatial dimensions, which is the X and Y, but you also have the ion dimension, which is basically if you're taking a slice of that cube, looking at one peak, and you're looking at the distribution of the intensity of that one peak across the whole tissue, that's basically looking at the X and Y for just one ion. So that's why it's called the data cube, because you have the X and Y, and then you have the different ion images, or thousands of ion images. So this technique is an untargeted analysis. It's mostly used for exploration, trying to find out what kind of molecules are there in the tissue of interest. You have varying different pixel sizes, which determines the spatial resolution. The most common with multi-imaging is 5 to 10 micrometer pixel size. You get up hundreds of molecules that are potentially identified, different metabolites, lipids, and drugs. And the typical size of the data sets ranges from 1 to 100, and can go even more. It depends on your setup. And as I said, this is the major technology for spatial metabolomics. And in terms of workflow, where does this fit in, or at least where METASPACE would fit in, is usually when you have a data set, it goes through a series of transformation and processing. So first you start out with the signal processing. So once you get the data from the instrument, you have to do some pre-processing before you can start looking at what kind of molecules are available. So you start out with signal processing, you do some normalization, centroiding, which is also otherwise known as peak picking. You do some correction to remove any kind of noises, you do some alignments, and then you look at certain things you overlay it with the microscopy image if you've already captured or acquired any microscopy image. You can overlay different kind of images as well as peaks. And then you can do some statistical analysis. For example, spatial segmentation, data representation. We'll talk more about this later in the end. But where METASPACE fits in is molecular identification. So this is the bottleneck and the major challenge in spatial metabolomics, is finding out what kind of molecules do we have in our tissue, because it goes from just having a bunch of peaks, M over Z's, into actual molecules that we can use to derive more biological insights. So there are different approaches. The most common is exact mass searching through libraries, trying to predict molecular formulas. And METASPACE is widely used as one of the most common annotation methods that helps molecular identification. And then once you have this, once you've used molecular identification, the next step would be some sort of validation to try to do like bulk LCM SMS, targeted MSMS, and some enrichment analysis, and some molecular filtering. So this is also follows up after the molecular identification. So what is METASPACE? Again, if you already know METASPACE, I'm sorry in advance. I'll try to be quick, but hopefully you get to learn something different. So METASPACE is a platform. It's a cloud software platform that provides two things. First is a metabolite annotation engine for spatial metabolomics. So you basically provide your data set, and we tell you what kind of molecules, or try to annotate or identify the molecules available in data sets. And it's also a knowledge base of spatial metabolomics, which is a collection of different and thousands of data sets. So the way it goes is you basically have your samples, different kind of samples from different species. You go through your sample prep. You have different technologies, which is different combinations of ionization sources and analyzers like MALDI, FTICR, Orbitrap, DESI. And then you have your data sets, which is the mass spec images and the hyperspectral data set that I just mentioned before. You have an optical image, you have a bunch of metadata describing the experiment. And then alongside this, you also have different databases that are used for identification. So when we're doing the annotation, we're doing annotation against known databases like the HMDB, Swiss Lipids, KEBI, depending on what kind of molecules you're interested to find. And METASPACE takes all of this. So it provides you with an FDR-controlled way. So this is also what is very unique about METASPACE. I'll explain this if time permits, through a web app. So this is a web-based platform where you can browse your data sets, you can visualize your own images, you can look at your spectra, you can compare different data sets, and it's also a public knowledge of thousands and thousands of data sets. So having this kind of platform allows you to perform big data analysis with different downstream functional frameworks. If you're doing single cell spatial, for example, You can share it with others with ease, which I'll also allude to later down. And then you can combine it, the results with different software to improve statistics, visualization, analysis, and so on. So as I mentioned, METASPACE is used for metabolite annotation. And currently in METASPACE, there are two different models for annotation. One we call the rule-based, which is the first ever model that is based on both spatial and spectral scores to try to identify the molecules. And the other is based on a machine learning model. The rule-based model is a simple scoring that basically multiplies a bunch of spatial and spectral scores together to come up with one score that is used to rank different molecules and tells you the plausibility of which molecule corresponds to a peak in your data set. There are other approaches. There are MZ-based matching or exact matching, as I mentioned earlier, and there are other vendor-specific software. So METASPACE is vendor agnostic. It accepts datasets from many different vendors, including Thermo, Waters, Bruker's, and many other vendors. And it's not tied to any specific vendor, although there are specific rules for uploading datasets that are still vendor specific. The second part of METASPACE is the knowledge base. So one thing I want to get clear from the beginning, METASPACE is not a repository of mass spec datasets because there are other existing repositories where you can basically put your dataset in order, for example, for a publication. METASPACE also gives you that, but it's not a repository, but rather a knowledge base, just a collection of many different imaging mass spectrometry dataset that you can browse along with metadata and metabolite annotations that you can use to compare with your experiment. There are other resources. For example, you have the EMBL EBI metabolites. This includes multiple different studies which have experimental metadata that you can look into. There is a metabolomics workbench, I think from UCSD. This is also NIH funded. These are different repositories. However, these repositories are not built for imaging mass spectrometry. They're mostly for bulk or LCMS-based mass spec. They do include, however, spatial metabolomics datasets, but they're not built for spatial metabolomics, unlike METASPACE. And throughout the years, METASPACE have had thousands and thousands of datasets. Here, if you're looking into both private and public datasets, we have almost 20,000 public datasets and approaching in total over 75,000 datasets. We have over 250 different groups or labs and over 1,500 different users. And the datasets span different polarities and different types of analyzers as well. I'm happy to go into the stats later on or also point out to the one on the side. So next, I want to talk about what you can do with METASPACE. So now that we know that METASPACE is a metabolite annotation engine and a knowledge base, there are various different things that you can do, various different features that we're going to look into during our live demo. The first is you can use METASPACE to do experiment planning. So before even submitting a dataset, one thing you should do is try to look into the knowledge base, trying to find what others found that has a similar setup to your own experiment. So the fact that we have a very rich knowledge base allows you to do so. And we also have a detectability app feature that allows you to know what kind of molecules you expect to find given let's say the matrix of choice if you're doing multi-imaging, for example. So these are kind of apps or features that can help you plan your experiment better. It doesn't do processing. So what you submit to METASPACE is not the raw output that comes out of the machine. You still need to do your own processing. So this happens outside of METASPACE. So anything related to peak alignment, calibration, normalization, centroiding. Centroiding is very, very important, which is the peak picking part, because you need to provide METASPACE with centroided data. So you do your experiment planning on METASPACE, trying to find out how to plan your experiment. And then once you get the data, you do the pre-processing actually out of METASPACE. Now, once you have the data set, there are a bunch of other things that you can do on METASPACE. I'll go through these briefly during the live demo, but usually you can do mostly visualization because METASPACE is known for its very powerful user experience and user-friendly interface because everything is on the web. You can look through your datasets, your iron images. You can do a lot of search and filtering by FDR, colocalization. You can do a lot of different spatial pattern analysis using regions of interest, enrichment analysis, comparison of datasets. I will not go into these datasets, these features more in details because that's what other webinars are for. Here, just introduction to what kind of features we have. And finally, Metaspace enables easy sharing and data of these data sets. So you can easily send what you find to a collaborator through how we actually manage those data sets. So how we manage them into groups, into projects, and how we allow the projects to be published and linked to your publication afterwards. And then how we can also export different images, intensities, annotated data, and also enable framework specific, like for example, data and so on. This is a more advanced feature that we can look into it afterwards. So before we go into live demos, I'll make a stop here to, if anyone has any question so far, if something that needs clarification, feel free to ask. Maybe you can take 5 minutes for questions. So feel free to raise your hands or also post on the Q&A. All right. Seems clear enough.
Seems there is like one question in the chat.
Sure. Which file from skills do you upload? Okay, so thanks, Georgia, for your question. So the what you upload to Metaspace needs to be imzML and the IBD file. It's not the skills workspace files that you have. You basically need to export it into imzML and IBD, and this is what you use to upload to Metaspace. And skills already have instructions to help you export your dataset into imzML. And then once you have it properly centroided, I'll also go through the instructions in the live demo. Then you can upload it to Metaspace. I hope that answers the question.
And just, yeah, adding here, we also have like on the documentation of Metaspace, all the instructions on how to make this conversion depending on like different vendors. I will post the link here. So if you have like any problems like on uploading or like the vendors not listed, you can like contact us that you can take a look into that. And it seems like Georgia has another question, Bishoy. So back to you.
Thanks, Lucas. I think we also have 2 questions. So I think Robin also asked the question, what kind of calibration you mentioned. So the calibration is part of the pre-processing before you actually do upload to Metaspace. So there is the peak alignment and the MZ calibration, which usually happens due to shifts in the MZ axis across the different pixels. We don't do that in Metaspace, but that needs to be addressed. For example, let's say if you're using Cardinal, they already have different tutorials on how to perform this kind of calibration before applying to Metaspace. But that's usually MZ shifts across different pixels. And regarding George's question about the animability, this is a very good question. So right now we do not support the animability data. We are planning, it's part of the roadmap to include it. So it's something that you can also add as a feature request. I'll also introduce what is feature requests. But as of this moment, we do not support ion mobility data. You can handle it with other open source packages, but we're planning to introduce it in the future. But currently it's not supported. Thanks for asking. Okay. Any other questions? All right.
I have a
So
question.
yes.
Sorry.
Yeah,
I have a question about statistical analysis. After analyzing the ion images for the particular one ion, for example, So how do you like start you're doing the stats on the
What
spatial
kind of
meta?
cluster are you looking for?
So for example, the region specific expression of the particular metabolite, if you want to compare, if you have one region which is having diseased, the one region is healthy, so we can see the difference. But
Yeah,
the thing is like we want to compare two or more groups in one slide. So how do you like
yeah.
see?
Yeah, so this I will probably address, and when I look into features, we do have this feature as part of Pro. So I will also show you that we have this during the live demo. I will show you where to find more information about this. But we do support this in terms of ROI differential analysis, and also a new feature coming up with Pro for cross-statistical data set. I think we're not going to talk about this right now because it's not the scope, but we'll be happy to chat about it afterwards.
Thanks.
Also, feel free to contact us after the meeting if it was not addressed further here. There's not a scope, but like if you contact us, like we can help you out with that.
Yeah,
Thanks, Lucas. Thanks.
awesome. All right, so I think we can move on to live demo. So let me, hope you can see my screen. Can you see my screen with MetaSpace? One second. Lucas, is it visible?
Yes.
Okay. So if you go to MetaSpace, this is what you see in the homepage. It's important to pay attention what you can do up top here in those headers. I'll just go over them briefly. So here, where you're gonna upload the data set, I'll go into this in a moment. This is where you can look into the knowledge base part. So different types of data sets, different annotation, what databases are supported and what databases we have, different projects that other people have submitted. There are different add-ons. These are also different features like detectability, converter, and Python clients. I'll also talk about this once we get to the docs. And here, this is the most important part, is the how to contact us, the docs, the frequent asked question, the feature requests, and how to learn more about MetaSpace. So one first thing that is important to get started, if you don't know anything about MetaSpace so far, is you need to first sign up to create an account. So if you haven't created an account, you should probably see that over there. So you'll see you have your accounts, your different groups, your different projects. And it's important to understand the difference between all of these. So there's a lot of information. And I am completely new to MetaSpace. What I do, first thing I do is I go to the docs. And I think this is the most important part. That's why I want to start here, because here you would know how to get started if you haven't dealt with MetaSpace before. So once you go to the docs, you'll see this Getting Started tab, different features, different guides. So here you're going to say Start Your Journey. And here there are different stages of how you can interact with MetaSpace. The first and the most important, which I recommend reading before getting started, is setting up your account. So you need to set up your account, identify what kind of groups and projects you have, and the API access. This is where you'll also find your API key. So what is the difference between groups and projects? So groups is basically the place where our different members. This is like a lab, for example. If there is a lab in a given institute, the lab has its own group. There are different members as part of the group, and those members, they have different data sets between them. So if I go to my account here and I go to this group, I call it, here I see a description of what the group is, what kind of data sets are shared that the members of that group can see. Here I can add different members to my group, which databases that I can upload that is specific to my group. So the group is just the place of your lab that's part of the institute, and everyone here sees the data sets that is part of that group. It's important to after you create your own account, you need to create a group as part of your institute. And then comes the project. So project is basically like a folder. So if you have an experiment, and that experiment constitutes different data sets, then you will create a project for these set of data sets, right? So each project has its own project manager, and you can also have members on a given project. It doesn't have to be part of the group, but different members, let's say different collaborators, can collaborate on the same project from different, let's say, groups or different type of users. But this is a different scope. So project is where you add data sets specific to a given experiment. And this is very, very important. I would also recommend you create the project before you start looking into other or uploading data sets. So as an example, let me show you the projects existing in METASPACE. So if you look in data sets and you click on projects, one of the most like number of data sets is the KPMP consortium project, which is a public project, and you can see these are different data sets, and there are different members of that given project. So again, a project is just a collection of data sets pertaining to a different to a specific experiment. So familiarize yourself with the difference between groups and members and projects, and then check what you can do in your account, because it's important to see something like called the API access. We're not gonna explain right now how to use a METASPACE programmatically, but when you need to do that, this is where you're gonna get it from. All right, so now that we know and understand the difference between the account, what can we get from there, groups and members and projects, the next thing is you need to prepare your data. So before you upload any data, you still don't know what to upload. I explained earlier that there are this detectability app. So if you want to know what's possible or detectable before you run your experiment, you would go to the detectability app, which is a different add-on, and then find out how to basically use this. So this is where you know how to know where it's detectable. And as Lucas already explained, in order to export your dataset into imzML so that you can upload it to METASPACE, we have a guide called Exporting to imzML according to different vendors and different configurations depending on which processing software that you used to help you export your dataset into imzML. So check this out before you get started. So we covered, now that you have the accounts, the groups and members and projects, and you know what to expect from your data, and now that you have your imzML data, the next step is uploading your data set. So before we go into the upload page, I just want to explain how the docs is organized. On the left, you see here two different panes. You see the feature panes and you see the guides panes. So the features is basically telling you, grouped by what kind of feature it is. It tells you four different things: what it is, when to use it, how to use it, and what the results would look like. So it's the same kind of structure for all different features. So if you don't know about any of the features before you use the feature, you would go here. Let's say, for example, you don't know how to share annotations. So you would go here, read about it, what it is, when to use it. I have this blocker right now, so that's why the video doesn't work. But basically, there's a video here, and then what the results would look like. And then you have the guides, and there are two different parts of the guides. There is the guide before submission, so you're already seeing how to export different imzML formats. But there's also a guide on how to use the upload page, which we'll go through right now. In order to make sure that you have a checklist of the files that you need, how to fill the upload page, what kind of information and metadata that you need, and so on and so forth. So we recommend reading the guide, especially this is also something that we're actively working on. The metadata recommendations is how to input this metadata. So now, without like stalling a bit more, let's look into how the upload page actually looks like. So if I go back to METASPACE and I click on the upload, so this is where you upload your data set. The first thing is you need to upload your imzML and IBD files, and then you have a bunch of fields to fill. The first is dataset description. It's optional, but I would highly recommend describing the dataset, especially for reproducibility later on when you're publishing this, because over time you're going to have many different datasets, and they're going to be in a project which should be involved in a publication, and someone else going to look into the dataset and they don't know what this is about. So it's nice to have a dataset description. And then you have the sample information of what kind of organism it is, which part of it, the condition, graph condition, sample preparation, tissue modifications, matrix, how it was applied. And then the MS analysis section is about mostly the instruments that you're using, the analyzer, the source, the different polarities, the resolving power. This is also very important to input correctly, because these numbers are used in the algorithm for the annotation. So if you enter a different resolving power or something that's incorrect, you're going to get unreliable or unexpected results. And then you get to choose which group this dataset falls under and which project. So that's why I told you to create the groups and the projects first before uploading any datasets, because once you already have the project, which is the folder set up, and the group, which is the lab or part of the institute, you're going to basically assign your dataset to the corresponding group and project. And then you have the quota and the visibility. This is part of the split between METASPACE and METASPACE Pro. I'll get into this a bit later. And then finally, you have the annotation settings. So the annotation setting is also very important, where you decide on which database, for example, you annotate against, what kind of adducts you expect to see, which model or version to use, whether there are chemical modification, what's the MZ tolerance, and the dataset name. So you'll see next to each of these are this helpful tooltips. You can just hover over it if you don't understand how to actually input any of these. Just look and they'll explain what these are. Okay, so and here you can look into the kind of different databases that you can choose from. So this is uploading the data set. Now, going back to the docs, now that you know, I'll also follow the METASPACE journey. So now that you uploaded your data set, you know the custom metadata recommendation, you know what kind of databases that you have. So also just to touch upon the database part. So you've seen from METASPACE here that you can select different databases. So these are the core databases that are supported by METASPACE, but there are other databases that are provided by the community that are called custom databases that are made public. And if you want to look into these, you're going to go into datasets here and look at databases. And when you open this, you're going to see a list of the available databases as core that is curated by METASPACE, and the community contributed databases that you can also download as a CSV file, and you can also use as part of your upload, right? So here. So you will see here the core, and then all of these are the custom databases that are provided by the community.
Also, just jumping on that, Bishoy, is also possible to upload your custom database without making public if you want. You can upload the custom database just for your group or just for yourself in case you're just testing and don't want to be public.
Yeah, exactly. So if you go here, also in the docs, click on the custom databases, you'll see a guide on how to prepare your custom databases, how to upload it, a video, sorry again about the blocking. And yeah, I'm happy to do it. Right now, I'm not gonna cover the details just for the sake of time, but it's important for me to know how to follow learning about METASPACE. It's basically following this journey, right? So getting organized, uploading, preparing. So now that we know how the custom databases, the metadata, the upload, for me, that's the most important part, is understanding the annotation page. So let's take an example data set. So here, I actually like this data set. So this is called the data set overview page. where you basically look at a summary of one of the data sets available in Metaspace. So if is how to reach there. So if you click, for example, datasets, you'll see a bunch of datasets here. Some things are currently processing, and you see this card in an interested dataset, and you click on the dataset overview, you'll get to the dataset overview page. So let's look at one of this datasets. So this is a mouse brain dataset submitted as part of the Mouse Brain Atlas project, which is a public project. And if you go look into it, you can see the publication DOI and associated datasets. So this is a lipidome atlas of the mouse brain. And in the dataset overview, you'll see the metadata, which is what we saw in the upload page, and the project associated with. And the most important part is the annotations. So you will see that a database is used, the number of annotations at different FDR thresholds, and the total number of annotations. So I'm going to click on Browse Annotations, which is the main page that you're going to spend most of your time on in Metaspace. So it's important to understand how to navigate this page. When you see the annotation page, you'll see the annotation table on the left. You'll see the ion image viewer, which is displaying the ion image, and then a bunch of other tabs like molecules, co-localized annotation, diagnostics, and metadata. Let's start with the table. So the table shows you the formula for any given annotation. And as I go through the annotations, it renders the ion image of that specific formula. And if you look at the ion image, this number here is the pixel intensity of that given annotation in that pixel. So you can see all these numbers, and you can scroll through different number of ion images that are annotated. So what you can see from the table is multiple different columns. So here you can see the ion image formula. Then you see the M/Z, the mass error, which is the difference between the observed and theoretical, the off-sample percentage, the MSM score. So I told you before that we have two different models. One is rule-based and one is METASPACE ML. The rule-based results in this something called the MSM score. And the MSM score is simply a product of these three scores. So it's called spatial, spectral, and chaos. These scores are from 0 to 1, and 1 being best, better, and 0 means worse. So if it's a good quality annotation, all of these scores will be closer to 1, which you can see it is. And the product of these three different scores constitutes the MSM score. Sorry, right here, right? And this MSM score is the score used to calculate the FDR, which is what you see here in the last column of that given annotation, which is the false discovery rate of that annotation. And then next to the MSM, you see three different columns describing the ambiguity associated with that given annotation. So you have isomeric molecules, isomeric ions, and isomeric ions. So isomeric molecules are these guys, right? So if basically it tells us that this annotation has 26 possible different isomers. The isomers are not dataset specific. They are related to the database of choice, which means that the M/Z of that ion can have these possible different molecules based on the database. So these are called isomeric molecules. So 26 different isomeric molecules. I'll skip the isomeric ions for now. The isobaric ions, which is here, these are formulas or other ions found in the dataset that have different molecular formula, but same M/Z. So you can see here that these two ions are isobaric to each other. There are different formulas, but very similar M/Z. So these are isobars. So again, Metaspace flags this for you, and it tells you that this annotation has two different isobars, and it tells you that's, oh, I clicked on one of them. Okay, let me go back. It tells you that this is one of them, which is here 10% FDR, and there's another isobar at 5% FDR. These, as part of the ambiguity, cannot be resolved, but Metaspace flags it for you to tell you, okay, this has this kind of ambiguity, and it's important to understand which is which. So this is what you can see in the molecules panel. And in terms of other columns that you can display, if you click here, you can look at other stuff like, what is the adduct, which is also the same here as you can see in annotation, but separately. Dataset and lab are relevant if you're having different datasets into one table, which is possible, but not right now. And then you have the database if you also have different annotation databases. So here, so right now I'm not applying any filters on the databases, right? You can see here those filters. So I can basically select a given database. So here I'm activating annotations of only HMDB. You can see here we only have 33 annotations that passes the 10% FDR threshold from the HMDB database. I can go to another database for Metabolome, has 182 different ions, also passing the 10% FDR threshold. And you can play with the different thresholds, but we usually recommend looking at the 10% FDR threshold. And then other filters that you can apply is you can look by a given molecule. So for example, if I want to look only for, if you have a molecule of interest or a formula of interest, you can paste this here, and then it will look through it and then load the different, here there are 3 different adducts of the same formula. So you can look by molecule as a filter, and then you can also look by adduct. And then you have this off-sample annotation. So I don't think this data set maybe does as off-sample probability. Yes, that's good. So off-sample basically, let me show you a good example of the off-sample. Yes. So these are all, we can see mostly empty ion images. Yeah, so for example, if, I mean, this is not off-sample because I'm mostly off-sample. Let me show you here. Features. Results. Yes. So if you look at this image, for example, here from similar data set, this is what's being classified as off-sample because the intensity of the ion, the selected ion, falls completely outside of the tissue, right? We already have a machine learning model that can flag this. So the off-sample that you see here, percentage basically gives you classification of what's the probability that this ion is actually off-sample, which is not part of the tissue that you have focused. And then what other stuff that we can look at? Yeah, so these are basically the filters and the tables, and then the ion image viewer. You can do different things like normalization, scaling, ROIs we're going to cover later on. And then you can look at the diagnostics, which is basically interpreting these scores, like the spatial, spectral, and chaos. I think we might be running out of time, so I'll probably not going to go into details of how to interpret this. We also have a separate webinar for these kind of features. But the take-home message is, if you don't know how something works, you go into the docs, read the corresponding feature guide, and then if you want to understand how to interpret it, you can go read the corresponding guide for interpreting those results. So I think that's good for the live demo. And wrapping up, because I also want to leave some time for questions, is now that you know what METASPACE has, or understand METASPACE, let me just spend a few minutes on explaining the difference between METASPACE and METASPACE Pro. So earlier in the presentation, I told you that the METASPACE platform earlier in last November split into academic and pro. We've went through what you can do with METASPACE Academic. The main difference is METASPACE Academic is for free and public data sets. So if you submit your data set and you are ready to make it available to the community publicly, you don't have to pay for anything. You will get it annotated as long as you make it public. While METASPACE Pro has the same capabilities of METASPACE, but is only for private, secure workspace. So this is with the part that you're going to have to pay for. And if you pay for METASPACE Pro, there are additional features that you get that is not available for METASPACE Academic. These features are mostly downstream features. So one of the questions that were raised earlier on is how to compare statistically for the same data sets or across data sets. These features are what we call downstream analysis, or more functional insights that you can get from the data. This is only available in Pro. So for example, we have ROI differential analysis. If you have one data set where you have tumor region and non-tumor region in the same slide, one data set, and you want to compare statistically the difference of what kind of metabolites are different in these two regions, we have a feature called ROI differential analysis, which you can go try it out for Pro. We have untargeted spatial segmentation to create clustering of the data set that you have, of each data set, so that you can find out which of the pixels have similar molecular composition. And we're also preparing to release soon the cross data set statistical analysis. So this is for projects where you have multiple data sets and you want to compare these data sets in terms of experimental conditions, technical replicates, also ROIs as well, so nested structures, and much more. So this is the main difference between METASPACE Pro and Academic. It's private and it has exclusive functional annotation features. And since you stuck around, as we promised, that whoever attends the webinar live will get something, and we want you to try out METASPACE Pro. So basically, for everyone attending here, whether you already have Pro, or if you already have Pro, then you don't need this. But if you don't have Pro, we want you to try it out, try out the features, and for 1 month. So we're giving you this full access to unlock the METASPACE Pro features to try it out. And in order to do so, we do really, really need your help in filling this survey to help us shape METASPACE Pro. So right now we've been over 10 months operating METASPACE Pro, and we need some feedback. So we will send you after this webinar survey, very, very short, maximum 4 minutes, that you can fill. And once you fill the survey, we'll send you how to access or how to unlock the METASPACE Pro. So this is basically only exclusive for those who attended today. So I don't want to take much of your time because I want to leave some room for discussion. We will send you also the slides as well. If you have more information, you can contact us. There's different support pages, as I explained earlier on the website. You can submit your own feature requests. Please check the docs. If you have any questions that wasn't available on the Q&A or on the support pages, shoot us an email. And yeah. And finally, we'd like to acknowledge everyone, the entire team, and everyone who supported and contributed to METASPACE, and all the funding bodies that help build METASPACE. Thank you for attention. Now we can get some questions.
So, Bishop, I have a question regarding your like you mentioned in the METASPACE, there are in one compound there are 26 features, like different isomeric compound. So is there any method or is there any selection criteria to prioritize which compound we can select on the basis of score or anything? Because in case of lipidomics, there are a lot of lipids having isomeric compound. So in that case, what should we do?
Yeah, thanks for raising this up, Raman. So one thing I tell you that you should not do 100% is, of course, you should not use FDR for this, because these isomeric molecules are based on the ambiguity coming from the database of interest. So if we're gonna put some filters on the molecules, on these isomeric molecules, it has to be filters or information coming from the database itself. So an example would be, if you're using HMDB, you already have a status. If you click on each of these molecules, you will go to the page for that given molecule. And if you look at the metadata on the HMDB, it can tell you, well, this has been detected and quantified. This has been detected but not quantified. This is expected, or this is predicted. So this is one of the filters, for example, that I would use to select or prioritize the molecules. METASPACE returns everything related to that IN formula. So out of those 26, what I would do is I would look into, based on the database information, not from Metaspace, what kind of information I can use. Something that is endogenous versus exogenous, for example. If something is found in an exogenous source, then it's probably not very useful to, your tissue of interest, then I'll probably filter that out. So anything for those isomeric molecules, any information from the database itself that you can apply to this would help you filter out or reduce this ambiguity further.
Okay. Yeah. Okay. Sounds good.
Yeah.
Thank you.
Yeah. Okay. Any other questions? Something that is not clear? Concerns?
Not, can I ask you another question for that?
Yeah, of course. Yeah.
Yeah. Like FDR selection value, like you mentioned that there is we should not like you are recommending 10% FDR, right? So in case of low abundant compounds, because we already working with some compound which is very, very low abundant in our imaging. And would you think like can we increase the FDR percentage for that, like 10 to 20%, or we should not go beyond 10%?
Yes. Okay. Okay. Thanks for the question. So the FDR percentage of 10% threshold is a general recommendation guidelines that you can follow, but it's not strict, right? So sometimes you find annotations that have FDR 20%. That is per your evaluation and assessment is actually a real annotation that you expect to see, right? So then you should not stick to the 10%, but then you need to explain why you consider this as a real annotation. The FDR is in comparison to like unplausible compounds, and it's affected by many different resources, the size of the database that you're using, the number of products and so on. So there are many different comparisons that affect the FDR. So it's not a set in stone threshold that you should use. It's a general recommendation rather. So, but you need to look at other different scores. So if I look at something that's 20%, and then I look into the diagnostics and I look at the spatial score, the spectral score, the noise, the MZ error, I need to check does it satisfy all of this? And if it does satisfy and it does have high score for all of this and still gets FDR more than 10%, there's something wrong, so maybe you can flag it and also you can send it to us and you can say, okay, why am I getting very high FDR with this? But there is definitely something that went off of the scores. You might prioritize different scores differently. That's why I'm saying your own evaluation beats the FDR, but you still need to do it in an informed manner through the scores.
Like other parameter, we can also cross check. Yeah. Okay. Thanks. Thank you.
Okay. Lucas, do you wanna say anything that I forgot?
Yeah. So thanks, Bishoy, for the presentation. Thank you everyone for attending. It was really nice to have you all here. So just a reminder, as you said, like this is going to be like a series that we try like each month to go one block at a time on how to use MetaSpace on your day-to-day work, right? So this was like the how to start, the introductory part. And on the next, we want like to go deeper and deeper on like the functionalities we have on the website. So for the next webinars, please tag along, invite your friends from like your colleagues from your groups, right? And each of like these presentations will try to give you some kind of extra resource like access for the pro futures or private data sets. So come like prepare your questions that we are here to help you. So as Bishoy said, you can find the resources on how to contact us on the website. So if you have any doubts like regarding downloads, analysis, how to use, feel free to contact us. We are here to help you. And we are in this process of building the community. So like always that also you want to give a feedback, you want to request a feature, we have the feature request page that Bishop attached. Feel free to do that because like we can develop the community with the help of the community. So like you guys are really important. And overall, that's it. And again, thank you for attending, Bishop, for the presentation. And I hope to see you guys again on the next webinar that probably is going to be like in 1 month. And don't forget to answer the reply to the survey. And then I'm going to enable the pro feature for you guys.
Yes, please. All right. Thank you so much for attending, and we'll see you around hopefully next month for the next webinar. Have a great day.
Bye-bye, everyone.
Thank you. Thank you, Bishoy and Lucas.
Thank you, guys.